All right, we're gonna start chapter 9, the cell cycle. But we're also going to be discussing how eukaryotic and prokaryotic cells divide. Now, when we talk about our prokaryotic cells include bacteria and archaea. These are going to divide by a process known as binary fission. Now, before we can talk about binary fission, we need to talk a little bit about prokaryotic cells. When we learned about prokaryotic cells, we learned the organisms, but we also learned about the location of chromosomes. Now, chromosomes in a prokaryotic cell are located in the central region of the cell known as the nucleoid. So these are the chromosomes. This is where the genetic material is located. It is always going to be found in the nucleoid. Now, when we look at the genetic material or the chromosomes, it's super condensed and it just looks like a bunch of big circles. Now, if we untwist it, the bacterial chromosomes, which is going to contain the DNA, we would say that the chromosome is going to be one large circle. So you could draw it like this. You untwist all the chromosomes, and when you untwist it, you have a single circular bacterial chromosome. Now, before a cell can divide, it has to basically replicate itself. So, when we say chromosomes, this is the area where we are going to basically contains DNA. So, chromosomes are going to contain DNA. So, to take DNA and copy it, we need to have a place to start. So, we have a region on the circular chromosome where we start to divide. This is called the origin of replication. When you talk about the origin of replication, this is the part of the chromosome that is going to divide. When you use the word replication, you are going to make a copy of DNA. So the origin of replication is where this is going to be copied. So what this looks like in a bacterial cell is it has to divide. When you say the word binary fission, that means 2. So here you have your bacterial cell. You need to replicate or make a copy of your single circular DNA. Now, your origin of replication is going to be where this replication is going to start. So I'm going to use blue to show that we are replicating the DNA. So what happens is it replicates its DNA, and now you have 2 sets of DNA. Now, the cell has to divide. Binary fission is basically going to create a new bacterial cell. So, when we say that the bacteria divides, that's after replication, but it divides by binary fission. So, let me pull this paper up a little bit. In binary fission, it starts to pinch in half. It's trying to divide. So the DNA has to let itself go, and it's going to have to separate. So over here, you have your old bacteria, and to the left, you have your new bacteria. So it's dividing by binary fission. So it starts to cleave itself until it can divide into 2 cells. So this one will divide, and it'll be a new cell. And this one will divide, and it'll also be a new cell. This is the process of binary fission. Now you have 2 new cells.